Joanne?
Hi, I'm Joanne. I'm one of the research nurses from the Newcastle Trust.
Great, thank you. Michelle?
Hi, I'm Michelle Edwards. I'm the clinical trials pharmacy technician at Freeman.
Thank you. Stephanie?
Hey, I'm the data manager on the surgical team.
Thank you. Ben?
And I'm the clinical trials coordinator on the team.
Thank you. Julia?
I'm Julia Scott, one of the research nurses on the research team.
Thank you. Oh, sorry, I didn't realise there's two Bens. Ben Cohen?
Hello, I work with Michelle in the pharmacy. I'm one of the pharmacy support officers.
Thank you. And Malcolm?
Hello, again, I'm a clinical trial support officer working with Michelle in pharmacy.
Yeah, thank you. I mean, it won't take long. We'll do our introductions and then we can always repeat ourselves. So yeah, I'm Chloe. I'm the trial manager for the UK arm of the study. I work at the Bristol Trial Centre, University of Bristol, and I work closely with Leah.
Hi, I'm Leah and I'm the deputy coordinator on the study.
And then Mark.
Hi there, Mark Edwards. I'm Prof of Anaesthesia in Southampton, so I'm acting as the UK CI for the LOLIPOP trial.
Thank you. We will just hang on for the PI to join before we start because we do need them present. Tell you what, I'm gonna share the slide, but basically when I share the screen, I can't see anyone's faces. So while we're waiting, I'm gonna see if I can work it out so I can see everyone while we've got a bit of time. There we go. Is that still just showing the slide? The first slide?
You can see this slide and the people on the call across the top.
Okay, fab.
Yeah.
And could you send him the link again, just in case he's having trouble logging in?
Yes, I will do.
There we go. Hi, Jonathan.
Hello, I'm just also logging in my computer, taking so long. I thought I'd just not hold people up and log in here, and then once my computer's sorted, I'll join on there.
Okay, great, thank you. We've done a quick round of introductions, and just to say that we are recording the SIV to include a video link in your SIV report. If that's okay with you.
Absolutely.
Great. Should we get going then, Mark? Can I hand to you?
Yeah, sure. Welcome, Jonathan. I think we've done some intros, but just briefly, I'm Mark Edwards, Prof of Anaesthesia in Southampton, so acting as the UK CI for LOLIPOP, and ask Chloe and Leah to introduce themselves when they take up their slides. So we're here to do the site initiation visit for your hospital for the LOLIPOP trial, which is looking at long-term outcomes of lidocaine infusions for post-operative pain after breast cancer surgery. Trial originated from the ANZCA network, Australia New Zealand College of Anaesthetists Trials Network, very well-reputed trials group who've done a lot of very big, impactful perioperative trials over the years. Invited us to participate with UK participants a couple of years back, which we accepted and managed to get some NIHR funding to run in the UK with the plan that we will recruit 1,000 of the 4,000 international target. So the trial is sponsored by my employer UHS Southampton, and we're very well supported by the Bristol's Trials Centre for trial management and so on. Okay, so next slide just gives a bit of a trial intro, which we've probably covered a little bit of it, early feasibility chats, but we'll just refresh. So we're looking at the effectiveness of lidocaine infusion given during surgery and up to 24 hours post-operatively, though probably not for many UK participants, and seeing how that affects the incidence of moderate or severe chronic post-surgical pain 1 year after the index breast cancer surgery. Next slide, please, which takes us onto the patient group we're looking at here. So quite broad inclusion criteria, essentially any adult female patient undergoing pretty much any primary breast cancer operation. So it can be mastectomy or more breast conserving surgery, plus or minus auxiliary components, can be unilateral or bilateral, and can also include simple sort of reconstruction done at the time of the surgery. So implants, very simple perforated flap, that sort of thing, but not very major autologous flap operations. And the patients must be ASA score 1 to 3. That leads us on to the exclusion criteria. We won't list them all out here because there are quite a few. Certainly, I'd say if you're looking at inclusion exclusion criteria, worth just having a little screenshot of those inclusion exclusion criteria at your fingertips on your phone or something if you ever ask, so they're just right there for you to refer to. But essentially the main things are firstly to avoid any confounders that might affect the interpretation of the trial results. So if there's pre-existing chronic pain at the site of surgery, or if this is a second operation or they're going to have a second operation, so it's not so clear how lidocaine will have impacted the first operation, then those are excluded. And then really we're keen to have this as safe an intervention as possible. So there are a range of comorbidities which might increase the chance or the severity of local anesthetic toxicity. So electrolyte abnormalities, taking certain drugs, cardiac conduction defects, that sort of thing, that act as exclusions. And an important point here is that this is a pragmatic trial. We don't need you to be doing a lot of additional medical interventions or investigations rather to find those exclusion criteria. It's really just things that would have been turned up during routine preoperative assessment. So they don't need anything other than the normal pre-assessment that they would have had. So, for example, if you've got a young fit patient and nobody would have thought to do an ECG for that patient because it's not indicated, you don't need to do it just for Lollipop. Okay, so it's just whatever information you can find from routine clinical workup. Okay, next slide, please. So, overview of trial process. Am I right in thinking we're only going to be targeting day case patients at your hospital? Is that right?
That's correct.
Yeah, fine. That is the case in a vast majority of our UK centres, and that's very reflective of NHS practice, where almost all of this surgery is done as day case. We do have one centre, Belfast, that are planning to perhaps do a bit of overnight infusions as well, but most other people are just doing the intraoperative stuff. So green is the sort of the patient identification approach and consent. The protocol is quite broadly written so that there's a range of ways that you can approach patients, depending on whether they're coming into the hospital for an appointment or whether it's a phone approach, that sort of thing. I think an important point really is that although the patient is technically allowed to sign the consent form on the day of surgery, we really encourage you to make sure that process where all the discussion and the thinking and perhaps the following up with the patient is done before the day of surgery. You don't want to be in a position where it feels like there's a question mark still hanging when you get to the day of surgery, because for the patient's point of view, that really feels like they're perhaps being put on the spot. And from your point of view, you won't have the certainty of knowing that they're definitely going to be included and be able to be randomised once they've signed off their consent. You can actually get the consent form signed in advance by posting it out if you'd like to do that. Or obviously, if you've got opportunities in a pre-op assessment clinic or something to get the signatures done, then that's another opportunity. There's a bit of baseline data to collect once they've done their consent, and then it's a case of preparing the intraoperative IMP ready for use after randomisation. So that we'll go through in a little bit. Randomisation must be on the day of surgery. Intraoperatively, obviously they get their CTIMP, so it's obviously the placebo or lidocaine. There's some data collection from the intraoperative phase. Bear in mind that will be coming from probably handwritten anaesthetic charts, so worth doing certainly as either at the time or as soon as possible after the anaesthetic's been completed. So if you've got any queries relating to bad handwriting or total doses of remifentanil or anything like that, you can address them with the anaesthetist there and then. And then there's a little bit of post-operative data collection in the recovery ward. And we'll go through that in a bit more detail as well. Once the patient's been discharged, it's very simple follow-up. Anything that is patient reported, so pain scores, quality of life, that sort of thing, anything patient reported, the patients will be sent a link to an online thing, and they can basically log in themselves and report their outcomes themselves. So you don't have to follow up for that bit unless they don't respond to the prompts. So there'll be an automated prompt, there'll be some reminders. If they're not responding to that, we'll turn the patient over to you and say, can you just phone this patient up to find out what their patient reported outcomes are at these time points. And then for a couple of time points as well, there's a brief medical review just to see what other treatments they've had. But hopefully the follow-up process is a pretty straightforward and light touch. Okay, any questions on the sort of general overview or in fact the trial overview training module? Hopefully we're doing this SIV on the basis that you've had the opportunity and have accessed the training materials, and this can be a bit more of an interactive sort of Q&A type session. So any questions on that or the trial overview training module that you've seen?
If they haven't got access to electronic to an email, can they still take part in the trial?
Yeah, absolutely. Yeah, it would just mean telephone follow-up. Okay. Right. Okay. If there's no other questions, I think we'll move on. And I think I might be handing over for a few minutes.
Yeah, thank you, Mark. So yeah, just a bit of a trial update. So obviously this is an international trial, and there are currently 18 sites open in Australia, and they've recruited 598 participants, 3 in New Zealand, and they've recruited 92 participants, 3 sites in Hong Kong that have recruited 349 participants, and then we in the UK have got 7 sites open and we've recruited 83 patients. So yeah, so as Mark said, this is sort of based on the assumption you've watched the training videos. So if you do have any questions sort of on screening, consenting and randomizing, you know, please do ask away. But yeah, we just try and make this less us talking at you. So yeah, just going to discuss a few sort of process questions for how things, how you see things working at your site. So first of all, could you sort of run through with us who you see will be sort of identifying patients and also approaching patients? So feel free to just shout.
I think we'll probably be identifying patients from the breast clinics originally, and we'll possibly go down and see them.
Yeah, so I think I suppose the surgeons will be the ones that will be highlighting the patients that are going to be done at the DTC who initially to us, who are the ones that will be eligible, as in, you know, the ones that we can, and then the research nurses will approach them.
Thank you.
And the surgeons are all, sorry, I was going to say that they're all very on board with it, so they're enthusiastic about it. So
great, that's good to hear. Thank you. So yeah, following on from that, then, could you run through your plan for day of surgery? Obviously, there's a lot going on the day of surgery. Plus, you've got to randomise, get your IMP. Like, how do you sort of envisage that working at your site?
sorry, I'm not quite sure which of us is going to answer that. Is it me or the research nurses?
It doesn't matter, to be fair.
We're hoping to start the IMP in the Freeman Hospital at the DTC. So once the patients sort of verbally agreed or consented to take part, we'll see them the morning of surgery and make sure that they're willing to participate and want to proceed with the study. And then we can, once we've either consented them, if they haven't been consented, we'll randomise them and get the IMP at the site, and 2 nurses will draw it up, and then we'll approach the anaesthetist to let them know.
Great, thank you.
So we're not going to have the first patient on the list because of the problems with, well, not problems, but having because we're doing it at the DTC, which doesn't have its own pharmacy. We're having it so that we can have the time to prepare the IMP. So it's not the first patient on the list. So there'll be time to consent, go through the baseline data and stuff as well.
Okay, so just from my understanding, the Freeman is separate site to the DTC.
It's a separate. It's a wing, but it's not connected by a corridor. If that makes sense.
Okay.
It's essentially in the car park. There's a 4-theatre suite.
Mm-hmm. Okay.
That's correct, isn't it, guys? I've not just spoken out of turn and said historical information. Yeah.
No, at the present time, we're hoping to. We've got a cupboard that's on order to store the drugs in the DTC itself rather than the pharmacy. So we're able to draw the drugs up, get them out the cupboard straight away and draw them up. That's still within the plan.
Yeah, great.
Great. So we might be able to include the first patient potentially on the list if once we've
Yeah. So yeah, logistically that will be easier. So yeah, sounds good that's sort of some future plans. Thank you. So yeah, next sort of discussion point. So this wouldn't be something we'd advise considering from the get-go. So like obviously familiarize yourself to begin with, one patient off one list, but have you thought about the potential sort of once you're familiar with the study of having 2 Lollipop participants on the same list and how you'll manage that, or whether you think that'll be completely impossible? Like, what are your thoughts? Maybe hard to say when you haven't begun.
Yeah,
but
it's hard to say at the moment, but they do have a long list and they do operate from 7:15 to well into the afternoon, so it might be a possibility, yes.
Great. So yeah,
You're
as I
only
mentioned,
providing one pump though, aren't you, at the moment? So it'd have to be like two like a time scale
Spacing.
between the patients, yeah.
Yeah. So yeah, so I was just gonna highlight that when you do sort of feel comfortable with the trial, you might potentially look to do more than one participant on the same list. Yeah, obviously you do only have one pump. So either you can make sure there's that time between the two, or there is also the potential to once the pump is in the mils per hour infusion, you can just swap to a standard clinical pump, which would then free up the LOLIPOP specific pump. So yeah, not to consider in the first, when you're getting familiar with it, but once you're familiar, something to consider to help your recruitment later down the line. So yeah, next question is just about sort of awareness and support around your site. So are attending anaesthetists, surgeons, recovery nurses aware of the trial, and have you got support from those staff groups? Obviously, they're quite key in the patient's breast care journey.
The unit's quite research active, and they're happy. We've visited the staff a couple of times, and they've been very favourable, and they're very open to research.
Great.
Yeah, I think that's pretty fair. And Exton, who's the charge nurse there, is very enthusiastic. I've been in email, quite a bit of email conversation with him. All the anaesthetists that work there regularly that do the breast list there, I've spoken to, and again, they're enthusiastic and keen, as are the surgeons.
Perfect. Great.
In
Thank
terms
you.
of, sorry, I was going to. I haven't provided them with training material as yet, so I think we probably need to do that.
Yeah, I was just going to say about that actually. So we have got 2 slide sets that you can use. So one for recovery staff and one for anaesthetists. So anaesthetists don't necessarily need to be on the delegation log. So if they're just like sort of the attending anaesthetists, they're not getting involved in the research any other way. They're purely the attending anaesthetist on a LOLIPOP case. They don't need to be on the trial delegation log, but they must have done that basic slide set that we'll send you. If they are going to be more involved in the trial, sign off eligibility, things like that, then obviously they do need to do a little bit more in-depth training and be on the delegation log. But we'll send those slide sets after the SIV for you, and you can just a little bit of localization to fit in with practice at your site, and then you can use those. And then last point is just have you had chance to use the staging database, which is like a test mimic of the real database, and do you have any questions about any of the processes at all?
So I did the training I did the training over the last week, and that's the one thing that I didn't have a chance to do yesterday. So I haven't logged on, but I'm aware that I'm supposed to log on. So I will. I don't know if the research guys have had a chance to do what I should have done.
Anyone had a little play around? Just say if you haven't activated your account on there, the link does expire. So we might need to send you the link again if you've not activated your accounts.
Yeah,
I've just found the email, but I've not been on it yet because I've been on annual leave. So I'll have a play around with it this week.
We'll resend those links then. Great.
It looks straightforward from, I have to say, from the videos. It looks relatively intuitive. I don't know if that is that fair. It looked like when they were talking you through the different steps, it looked like that's how you'd expect to use it.
Yeah, it is. Yeah, it is pretty simple. It just allows you to familiarize yourself with how to, do the whole process. And in particular, we'd say, have a gut randomizing a patient and also unblinding a patient, just so you're familiar in those situations. If you did need to unblind, hopefully you won't, but it could be a bit heated. You'll know exactly how to do it. It's just, familiarizing yourself with that process. If there's no other questions on screening, consenting and randomizing, I'll hand back to Mark.
Great stuff. Thanks very much. So just focusing on a couple of the bits that will be a feature of the day of a LOLIPOP operation. So one of the main sort of talking points is the dispensing sheet. Once you've randomized, or rather as part of randomization, you have to put in the patient's height and weight and so on, so that it can calculate a lean body weight for you for that patient. So that'll be calculated for you. And then when you print the dispensing sheet, that'll be put on there. And then that forms the basis of doing your vial cross checks and the cross check for the pump setup. And it also gives you some useful bits from the protocol around parallel local anesthetics. So if other local anesthetics wish to be used either for blocks or wound infiltration, then the maximum permitted doses for that patient, knowing their weight, will be printed on that sheet. So it's a nice personalized sheet for that participant. Useful. I recommend our research teams take these along to the team brief for a particular list, to basically say, right, we've got this patient coming in, just to flag awareness that they're in Lollipop, and also to discuss with surgeons and anaesthetists, if they do want to use other local anaesthetics, how much will be allowed to be used. So that's there in blue. Obviously, it's either or. It's not all of those alternative local anaesthetics can be given. Additionally, it's a case of choosing the one that they want to give. And then in terms of cross-checking, the first stage of cross-checking is getting the vial out of the cupboard, and that's research staff member number 1 and 2. They check with each other to make sure the vial number is correct as per the dispensing sheet, expiry date, all that sort of thing. So quick signature there. Then the second cross-check point is after the research team member has set up has drawn up the vial and has set up the infusion pump. And once they've done that, they'll wait till the anaesthetist is in a good moment and cross-check that with them, and ask the anaesthetist to put their initials and sign off that they've checked that the rates that are given on the dispensing sheet are the same as what the pump has calculated that it is planning to give the patient. So there's that sort of that sort of calibration. Now, one very minor detail that some eagle-eyed team members have noticed at some of our sites is that occasionally there'll be a 0.1 mil per hour discrepancy between the pump and the dispensing sheet on the bolus rate. I think that's just due to the rounding, the way the rounding happens in the two ways of calculating it. I'm sure you could do the maths and realise that a 0.1 mil an hour difference in the rate essentially is completely insignificant. So it's just a very minor mathematical rounding difference. So you're okay to sign that off as being the same if it's just 0.1 mil an hour. Okay, next sheet, please. And this just takes you through a little bit more about the Alaris pump. You might have noticed that some of the original Australian training materials talk about the Alaris PK. Since the trial's been going for a couple of years now, BD have actually updated that pump now to something they call a Nexus. It's very, very, very similar. Internally, it's got a few different features, but effectively, anyone that's used a PK pump will find this completely familiar. The one minor difference is it's programmed in a slightly different way, and we have put some new training video onto the sort of pump training to explain that when you're doing the cross-checking, you just need to pause at a slightly earlier stage of pump setup. So it's a very simple case. Turn the pump on, choose the Lollipop program. That's the only named program that will be available on the pump. There won't be any sort of total intravenous anesthetic models or anything available. It'll be reset just Lollipop or mils per hour. You put the patient's lean body weight in there, and at that point you'll get a confirmation screen of what is being planned to give for the bolus dose and the mils per hour rate, and that's the thing to cross-check against the dispensing sheet. You can leave it on that screen for as long as you need. It won't alarm and be irritating, and you can just wait until the anaesthetist is ready to cross-check. And once that's been done, you basically press OK, and then it'll be on hold waiting for the anaesthetist to connect it up to the patient's cannula and to press the green infusion button when they are ready after the patient's gone off to sleep. The other pump thing to mention is that they'll probably need a brief checking in by your medical engineering team. They will have already been programmed by our medical engineering team in Southampton, and they'll be ready for use, but it's quite regular for local medical engineering teams just to want to usually just electrical safety test them. And if the pump needs maintenance during the trial, we'll get it shipped back to Southampton so that the maintenance can be done there. They've got a 3-year life cycle for maintenance, so it could well be that if we go well, we might have actually finished recruitment by the time you need to send it back for servicing, all being well. And they're very reliable. They've been in clinical use for many years, these pumps. Okay.
Over to me again. Thank you, Mark. So yeah, just run through some safety aspects of the trial. So yeah, on the screen now you can see a summary of the study drug infusion stopping criteria. So this can be found within the protocol, and we'll also send you a printed copy in your trial startup pack. So you should keep this printout with the patient at all times whilst the infusion's running. So that includes when they move into recovery, and yeah, basically just outlines when you should stop the infusion in different settings. And then moving on to the LIST tool, so the lidocaine infusion surveillance tool. So this tool is intended as a rapid aid to identify clinical signs or symptoms that may indicate the patient starting to develop lidocaine toxicity, and then that will lead to you stopping the infusion. So there's 5 symptom questions and 7 clinical signs are assessed. You should start, so for day case patients, you should start completing the tool once the patient enters recovery, so within 15 minutes of their arrival in recovery, or when the patient is awake enough to sort of have a conversation with you. For day case participants, you should ask the 5 questions again once they meet the criteria for recovery discharge or stepping down to reduced monitoring. If at any point the patient answers yes to 2 or more symptom questions or has any signs or complications at any time, the infusion must be stopped. So after you stop the infusion, if the patient is otherwise sort of well and stable, you should sort of repeat the LIST tool hourly. If all symptoms are resolved or 2 hours have elapsed, then you can sort of stop monitoring them there. In this instance, no need to unblind the participant. If the patient, however, remains unstable once you've stopped the infusion, you should sort of initiate rapid or urgent review so however it works at your site, the anaesthetist, theatre emergency team, etc. Tell them the patient is a LOLIPOP patient and there's a possibility of lidocaine toxicity. If it is suspected that they're having severe lidocaine toxicity symptoms, then you should follow the AAGBI guidelines and unblind the patient. And then probably goes without saying, but if intralipid is a requirement, so for example, the patient's in circulatory arrest, don't wait for the unblinding to sort of initiate given that intralipid. All suspected lidocaine toxicity events are then recorded on our CRF form 13, and they'll need to be reported to us as the UK sponsor at your earliest opportunity, and this must be within 24 hours of the event happening, and that's by you completing that form 13 and enter it onto the database. So I think key points here is just to say that stopping the infusion doesn't necessarily mean you need to unblind. So this tool is intended as a bit of a safety net, really. We're trying to catch, we're trying to stop the infusion before, the serious events happen, essentially. So is there any questions at all about using the LIST tool?
Can I just clarify there? So we stop the infusion if you trigger on signs of local anaesthetic, of like early signs, but you only need to know within 24 hours about unstable LAST that basically requires intralipid and or resuscitative intervention. So slurring of nystagmus and those sorts of things that you might metal taste is not notifiable.
No. So yeah, you'll enter when you ask the LIST questions, that obviously then goes onto the database and we can see if the infusion was stopped from that. It's only if you then go on to require unblinding and there's sort of a medical emergency that you would report it to us.
And am I also allowed to ask whether that's happened yet at the UK? Presumably, I presume not. Or
So we've had one event. However, it was after a conversation sort of with the site team. It was more, might need Mark to come in on this clinically, but it was more a case of there was pre-existing medical sort of conditions, and it was more a case of wanting to unblind to know if it wasn't lidocaine toxicity, how they should proceed to treat the patient. Is that right, Mark?
Yeah, I think that's a good summary. It was definitely not a straightforward, easy patient. Oh, look, they've got some funny symptoms. This is, we're worried about toxicity. It was a very complex patient with a slightly funny presentation after an anaesthetic that had raised a bit of concern with the local clinical team. And when the case was reviewed with the local PI and the research team afterwards, I think everyone felt that the likelihood that had been local anaesthetic toxicity was actually exceptionally low. It really didn't fit with that picture, and they didn't have any of the markers of severe toxicity. They had not had any. They didn't have an arrest or seizures or anything like that. So it was just a bit of diagnostic uncertainty, really, in a slightly complex patient. And I think in Australia, obviously elsewhere in the world, over 1,000 patients have been randomized now. I think they've had definitely one, possibly two unblindings. And again, neither of those in the end were felt to have been likely to be a true local anaesthetic toxicity. So I think safety-wise, things are looking very good. I mean, I think the key thing is really the LIST as a screen is very non-specific and very sensitive. So, a bit of bradycardia that doesn't get better with a bit of simple treatment, that's it, it's stopped. There's a bazillion reasons for patients being a bit bradycardic. So it's not sensitive at all, but it means there's a very low threshold for stopping, just to keep the trial as safe as possible. And I think that seems to be working well. I think it's probably the right approach.
Great. I'll move on if no further questions.
And sorry, I had one other question about that. So us telling you that we've in the situation where we've told you within 24 hours, telling you is putting that on the database. Do we have to ring you or contact the study team?
I mean, it probably is good. So you definitely have to complete the form and enter it into the database or email it. Obviously, if that's an emergency, just get it to us however you can. But it might be good to have a phone call with us.
Is that an SAE form? Sorry, do you mean an SAE form?
This is actually a different form, but you've just made me think whether you would also need to do an SAE form. I'll have to check what we've put in the protocol, whether it would be SAE and this. I'm going to put that as an action to get back to you on that, if that's okay. I have a feeling it's SAE and...
Yeah, but there is a specific unblinding form.
Yeah. But yeah, it is probably good. Like the site who did unblind, like we did have sort of a bit of, I think it was email correspondence. They emailed us straight away when it happened. And yeah, we then sort of just arranged a call after to check, everything was all good and the patient was okay and things like that. So yeah. Great. Thank you. So yeah, on to unblinding. So yeah, some examples where unblinding would be required would be sudden unexplained loss of consciousness, tonic-clonic seizure, life-threatening arrhythmia, or cardiovascular collapse. After randomization, you have the ability to unblind on the database for 36 hours. So it's really important that if for any reason a participant's surgery date is changed, that you remember to update that surgery date within the database so you still get that 36 hours to unblind. Anyone with an account on the database will have the ability to unblind a participant, but also recognizing that this is an emergency situation and anyone needs to unblind, you will have a generic unblinding login. So again, in your startup pack, we'll send you a laminated copy of the unblinding guideline that you can see on your screen. So it's usually the lead research nurse will be sent a link to set up a generic unblinding login account. So you'll be given your username and you'll create a password. Just to highlight, that password should be something generic, not personal, because it's going to be used by anyone and it'll be written down on this piece of paper. Quickest way to unblind for anyone, even if you've got an account, is to sort of scan that QR code, go on the generic login, unblind. So just because you have an account on the database doesn't mean you need to use that to unblind. Just, yeah, whatever's the quickest way in that situation, you can unblind. And if the participant is unblinded, then we do have an unblinding CRF form. So form 11 will need to be completed. And then obviously also that suspected lidocaine toxicity form. So is there any questions about the unblinding process or anything in the unblinding training video? Yep, I shall move on. So there are some safety endpoints listed in the protocol, and these are sort of documented either within the CRFs, or if there's cause for concern, then they may warrant sort of SAE reporting, etc. So the study infusion should be stopped at any time if any of these events arouse cause for concern or they can't be resolved following treatment. So there's a bit of overlap in this, these safety endpoints in that table I showed you a few slides ago. So yeah, a lot of these events on here will be considered expected reactions and don't require expedited reporting to the sponsor, unless the nature and severity of the event is sort of inconsistent with the information about the drug in the reference safety information, which in Lollipop is the SMPC. So yeah, as I said, most of them are captured within the CRFs or on the adverse event form, but if there was, if it did meet one of the serious criteria, then it would then be reportable. Yeah, a bit about protocol breaches. So protocol breaches are reported on Form 10 of the CRF pack and entered onto the Lollipop database. Unfortunately, in the UK, there is also a paper protocol deviation assessment and report form that needs to be completed and emailed to the 2 email addresses you can see on the screen there. What I will say is if you have a protocol breach, fill in one of the forms. If you just complete the CRF form, we'll remind you anyway. So if you don't remember to complete both, it's not an issue as long as one's completed and we're aware of it. We can give you a prod for the other one. The Australian team have broken down some examples of protocol breaches within the protocol, but just to say that any other deviation from the protocol in the UK is a protocol deviation here, so it would need to be reported. Yeah, I won't sort of go for every single one, but just sort of one to pick up on is under the non-infusion related, there's the open label administration of lidocaine in excess of permitted amounts. So just to say, if that was sort of picked up, if the infusion hasn't been started, so if the trial infusion, hasn't been started, then we would say don't begin the infusion there, because obviously we don't know if they're going to then have lidocaine that's going to be given in toxic amounts. If the infusion has started and is picked up, we say stop the infusion again. We don't know if that patient's then having additional lidocaine. And then, yeah, just a bit of summary on safety reporting. So yeah, I mentioned the summary of medicinal product characteristics is the reference safety information in the trial. I'm not going to go through every form on here because you can refer back to this, or the CRF pages should be pretty self-explanatory, but obviously we have the standard AE, SAR, SUSAR, etc. forms, and just to say, obviously, any serious adverse events, reactions, SUSARs, safety issues, suspected lidocaine toxicity, etc., need to be reported to us within 24 hours. But I am sure you are all very aware of that. So is there any questions on anything sort of safety reporting related from either the videos or anything I've said today?
No, I don't think so.
Fab. And then my last slide before I hand over to Leah. So this is just to highlight a bit of sort of a quirk with this being an international trial, is that dexamethasone doses are sort of reported a bit differently between Australia and the UK. So essentially, this was picked up after the UK started, recruitment and entering this data. And it's been decided that as obviously the trial had already begun recruitment in Australia, we're going to go with reporting the Australian doses. So yeah, you can see on the screen there, if 3.3 milligrams of dexamethasone sodium phosphate used, it'll be documented as 4 milligrams of dexamethasone phosphate, and then same with 6.6 and 8 milligrams on the screen there. Again, if you forget this, because that's probably, there's a lot to remember anyway, and then this is an additional quirk to add. If you forget, it will be flagged as a query on the database and you'll just be asked to update it anyway. So if you can remember, great. If not, it'll be flagged anyway. So, but just to highlight, that's a bit of a glitch, I guess, that with this being an international trial and differences between the countries. And yeah, if you refer to the CRF manual as well, when you're maybe completing your first few patients, this is documented within that. So I will hand over to Leah now to go through some follow-up.
Thank you. So just to start by summarising the follow-up time points in Lollipop. So we have 3 follow-up time points at the moment, 3 months, 6 months and 12 months. So these are mainly patient-reported questionnaires. So at all 3 of the time points, we're collecting those. And we do this on a REDCap database, which is actually separate from the main database. And it will send out the questionnaires to the patients via email at the time points for you, and also send them some reminders by email and by text, so they can complete that all online on their own time. If, as we sort of briefly mentioned at the start, if the patient doesn't have an email address, or if they don't want to complete it this way for whatever reason, you can do those over the phone. The sort of reminders and things will just get sent to you as a study team to remind you to do those, which is quite useful. Just to note that there's also a bit of data collection from the medical notes at 3 months and 12 months. This data is actually collected on the main database. So if you're kind of not sure which data gets entered where when you start out, it's just good to refer to the CRF manual for your first view until you get the hang of it, just because we don't want people sort of having to re-enter data in the wrong place. Like, you don't need to enter the patient-reported questionnaires onto the main database. We'll upload those for you. And then finally, just a note to say that if patients miss their 3 or 6 month follow-ups for whatever reason, we do still ask that you attempt to collect that 1 year follow-up. Obviously, unless the participant's withdrawn, that's just because the 1 year is our primary outcome. So it's really important to try and collect that. Are there any questions on follow-up at this time? Okay, I'll move on then, please. Thank you. So this is just a note to say that we have a little guidance document, which is to help participants with calculating their total daily dose of pain relief medication. These are the 3 and 12 month time points only. So this document's available on the REDCap database. So when participants are completing the questionnaire, they can just click onto this to see it, and it will help them make those calculations. For any participants who have opted to do the questionnaires over the phone, we just ask that you post a paper copy out or send a copy out by any other means of this document to them ahead of their follow-up, so they've got a bit of time to read it before you talk to them. Okay, next slide, please. So we've just got a few discussion points now to kind of consolidate knowledge from the past few slides. So first one is just what you would do if a participant was randomized on the day of surgery as per protocol, but then their surgery had to be postponed. What are the things that you'd need to do for that?
So one thing you'd have to, oh, sorry.
No, sorry, you go.
I was going to say change the date of the surgery so that it allows unblinding within the 36 hours.
Yeah.
Would be one thing.
Yeah, that's the main one. So depending on how long it's been postponed, you might need to recollect consent. So I think we say if it's been postponed by more than 30 days, you'd also need to recollect the baseline information if that was the case. But anything within that 30 days, you don't need to do that. Also, just worth mentioning on the vial they're randomized to, so obviously that can just be used when they actually have their surgery, provided that it's not already been drawn up. So we just recommend that you sort of wait till the last minute to draw that vial up in case of any delays or issues. If the vial had already been drawn up and the surgery was postponed, then you would actually unfortunately have to dispose of that vial and they would not be able to go ahead with the study. Okay, so next one is around safety. What would be your procedure if the participant had a seizure in the recovery area?
I'm happy to answer, but I don't know if Julia's just unmuted. So
no, we just usually stop the infusion
Yeah.
when people have seizures or anything untoward.
and I think that would trigger a
like a risk, like an emergency
proper
response.
Yeah, exactly. Yeah, so urgent anaesthetic. They said it was the I can't remember if it was like a medical. It's like a resus team, provided that it's got an anaesthetist on, something like that. It says so. It would be in the reality, it would be an anaesthetist in the DTC and first responders to whoever that is there. I should just check if it's a normal Freeman arrest team that would go there.
Yeah, that sounds good. Yeah, and you also might want to consider unblinding.
Yeah, that's true.
And then just completing all the relevant paperwork for that. So it'd be suspected lidocaine toxicity as well. Yeah.
I think that if because that's pretty. If someone who you wouldn't expect has a seizure in recovery, is that something? Do we have to unblind, or is it? I mean, I suppose it does make a difference ultimately to whether it's how you then treat it. So it's probably is worth you definitely want to know, wouldn't you, whether you give them lidocaine or not. But is there a situation where you might not unblind? Is that up to your discretion? It just seemed.
No, I think we'd be looking to unblind really for all patients.
Yeah.
I think at that level you sort of need to know, don't you? Because
yeah,
if they're in the placebo group and they don't normally have seizures, then why on earth they had a seizure? So that would probably trigger a lot more further investigations than if they were in the lidocaine group. You could say, okay, well that was a lidocaine toxicity event. I think, are we recommending intralipid as well for seizures?
AAGBI.
Yeah,
so intralipid as well as part of the resuscitation.
I think the AAGBI guidelines, I think, say you treat seizures. I'll have to have a double check on the AAGBI guidelines would be you'd think you manage seizures as normal, don't you?
So just flip back a few slides to the list.
Yeah, I have to have a check. Yeah,
list ones, and we can just. Sorry, it mostly is for circulatory arrest, isn't it?
Yeah. That's
Yeah.
That's fine.
you'd probably be bringing it closer to the patient, just in case things progressed.
Absolutely,
But,
yeah.
Yeah.
Great, thanks, guys. So next one is, what would you do in the unlikely event that the pump failed during the bolus infusion?
So this is on the video that I've seen. I think you turn off the pump, I think, and then you say it's and then you turn it on at a mils an hour or something. Is that right?
Yeah, that's right. Yeah, you wouldn't restart the bolus.
Yeah. And then presumably that goes down then as a protocol breach, does it? Or we have to presumably record that it didn't give it properly.
Yeah.
And
I was going to say, Mark, you usually jump in with occlusion alarms and stuff.
Yeah, no, absolutely. So I mean, this is a very unlikely situation that the pump just completely blacks out, perhaps if it's been left to run down the battery a little bit or something like that and not plugged in. Essentially, if it's midway through the bolus, it's the real concept is not trying to recreate that bolus because you don't know how much exactly was given, so you just revert to the mils per hour. But if it's something straightforward, like, we know the pumps have got occlusion alarms.
Yeah.
If it's something very straightforward and it just temporarily halts, but it's still in touch with the program, and you can just, it's a cannula flush or straighten a line out or something like that, and it's able to be restarted with no problems, then that's fine. So you can sort of continue a bolus, but you would never let it restart one from scratch.
Great, thank you. And then probably one for the research team. What would your process be if a participant wasn't responding to their 1-year follow-up questionnaires?
Would a research nurse call the patient?
Yeah, that would be a good start. I mean, we generally say sort of 3 attempts to call them. Also, just to note at this point that we do allow the 1-year follow-up to be collected up to 15 months after surgery. So you do have quite a lot of time to collect that if the participant's not responding or if they're away for whatever reason. Yeah. Okay, next slide, please. Right, so just to start tying things up, after the session today, you can finalise your training. So we've got a training log on Microsoft Forms for you to fill out. There's a link there, or you can use the QR code, but I'm sure we've sent this to you before as well. So you just record there all the other kind of videos that you've watched, hopefully before this session, and also the session today. And then once you've done that, you will need to register on Mango, which is where we have our delegation log. It's an electronic delegation log platform. Yeah, sort of everyone in the main research team will need to sign up on there. I think, as we mentioned, sort of attending anaesthetists don't need to be on the delegation log, but yeah, so something to note with Mango is the way it works is every time someone registers, we'll sort of check that like they've done all the kind of training that lines up with the tasks they've assigned themselves, and then that will go for approval by the PI once they've registered. So as PI, you just sort of log on to Mango and you'll get a kind of a list of who's there for you to approve. And you should also get notifications as well each time someone sort of pops up as ready for you to approve. But yeah, we can provide a bit more guidance on that if you're not sure, but it is a very straightforward system. Okay.
Just to say, Leah, so I was just going to say to Jonathan, for the PI role, we do ask that you select every task, although you might not be doing every task, it just sort of shows oversight. So if you could select them all. Thanks. Yeah.
Thank you. So just a quick note to say that we are registered on the Associate PI scheme. Quite a few of our sites are taking part in this, and they found it really useful. So just something to consider if you haven't already. Next slide, please. Okay, so I'm going to take over control now just for a bit to run through the site checklist. Just give us an idea of sort of where we are in terms of setup progress. So obviously you've submitted the feasibility form to us. So next one, PICV and GCP certificate. So when you register on Mango, these actually get uploaded by the person creating the account. So we will get those anyway. The protocol agreement it's actually within the protocol, and that's just something that we need the PI to sign, please. And then if you return a copy to us. So training logs. So yeah, everyone needs to complete a training log who's here today. I think we've had a few come through from the pharmacy team already. And then, as I said, the delegation log needs completing. MNCA, I think I've seen that come through while we've been on the call, actually, for us to send out for signatures. So we'll sort that out, and hopefully that'll be going around soon. Is there any sort of update on where things are at with local approvals? Does that happen alongside?
Yeah, pretty much once the MNCA is being fully executed and signed off, the approval should come very quickly after that.
Okay, great, thank you. So next item is ISF checklist. I'm not sure if we've had that from you yet.
No, I received that yesterday, and I'm going through. I'm trying to create an electronic local site file on our shared drive, and I'm just going through the checklist one by one and adding everything that needs to be there.
Yeah, great, thank you. Okay, I will tick this one off for today. You said you've got your cupboard on the way for the IMP, so that's obviously in progress. The pump, so we will send that out to you once we've got the contract signed. So hopefully we can arrange that soon as well. We've already spoken about the staging database. We need to resend some links for that. And then the next one is just to confirm that you've got some 50ml syringes available. Yeah, cool. And intralipid, I'm sure you've got that available. Yeah. Localization details have been provided. We've sent the localized documents to you, I think today or yesterday.
Sorry, Leah, can I just check on that? We just wanted to check, because I think they're localized with Royal Victoria. That is the name of Newcastle. So, like, but they're going to Freeman. Like, if that's right, or they need to be localized with Royal Freeman or Freeman. Sorry, I might be getting the names mixed up.
And there's basically two sites. We've got the Royal Victoria Infirmary and the Freeman Hospital, but they both come under the same trust.
Okay, so they're localized at trust. Is that okay?
Yeah.
Okay, that should be fine then.
Yeah, so we should call it Newcastle and Pontine Hospitals, I guess, is the correct thing that how we do it, I guess.
Yeah, I guess if you just double check the localized details that have been sent, that you're happy with them, and if not, we can adjust.
I will do. Yeah.
Thanks.
Have you got access to the CRFs? They're on our website, so you can find those with all the other kind of training and things like that. Names and lots. Fab. We've already spoken about the ISF setup, and pharmacy site file will also need to be set up. We don't have a checklist for that, but we can provide a contents page if you don't already have that. Yeah, and we've already spoken about the training with relevant teams, and last one there, I'm sure you'll do that when you get access to the staging database. So, yeah. sharing that.
Great, thanks, Leah. I won't reshare the slides because the last slide is just our contact details, and you'll have a copy of those. So, yeah, I guess to finish up, have you got any other questions or anything for us at all? Anything we haven't covered today, or?
No, I don't think so. We just need to have a chat as a team, I think.
Great. Mark, is now probably a good time to talk about.
Yeah, just wanted to double check the sort of happiness of the local clinical teams with the sort of the parallel local anaesthetic dose limits. John, is there much usage of specific blocks, wound infiltration, that sort of thing? I think we had one that's open to recruitment that had a surgeon move in from Newcastle who was very keen on PEX blocks and didn't really want to limit their dose, apparently. But I'm not sure if that's a Newcastle thing or if that was a surgeon thing.
He was given the who was
So
the surgeon was given the block. No.
Was putting in the PEX blocks, PEX 2 blocks, and possibly even doing some wind infiltration as well. So sort of using the absolute possible dose basically. But.
Yeah. So it depends a bit on the case. There's so all of the major breast surgery, however you define that, gets a block of some sort, which is probably a paravertebral block, but is a bit anaesthetist dependent. And for the minor breast surgery, wide locals and that sort of thing, which is more likely to be in this because it's at the DTC, there's a variety of practice. So everyone gets local of some sort, but I would say it's about 50-50 whether they'll get a block or local. So that's whether it's a field infiltration by the surgeon, which is a type of block, or an anaesthetic delivered paravertebral or PEC block or whatever. So I would so one of the main things that we discussed as a group before agreeing to join the trial was whether we were happy with one, potentially withholding blocks, but actually the you can, and so I think yeah, so so the anaesthetists I've spoken to who were working there are all, well, we discussed it as a group and happy with that. I do think it's absolutely key that we communicate the doses that are allowed, but that is part of the study, isn't it? But and there's a lot about local anaesthetic in the current it's quite topical, isn't it? But I think communicating at the start of the case the milligrams that you're allowed is probably, we should probably do routinely anyway. But yeah.
So I guess getting that sweet spot, isn't it? Making sure people are happy that if they do want to put a block in, they're happy with the dose limit.
Yeah.
Similarly, not having people be too worried about using any additional local if they're just generally paranoid about locals.
Yeah.
But from your conversations, you don't think, it doesn't sound like that's going to be a problem?
No, I don't think there's resistance to following the protocol, if that makes sense. And there was a bit of a discussion about whether we thought that the protocol was acceptable in the sense of, like, firstly, what do we think about doing lidocaine infusions and what do we think about mixing them? But we decided that this has got ethical approval and it is evidence-based, and therefore if you're going to do this, then it should be within a trial, and therefore that's the place to do it, isn't it?
Yeah, no, that's great.
Yeah.
Handy statistic from the trial in general, obviously about 1,000 patients randomized, as I said, in total. About 60% of them have been getting additional locals, either block or infiltration. So, a majority of patients have been getting the additional local. I'd imagine most of those go up to the dose limit. And as I say, no solid events that indicate a local anaesthetic toxicity at any stage so far. So
I think that's just one thing on the two different form, the form that prints out the doses and the infusion rate and all those sorts of things. On one it said levobupivacaine, and one it says bupivacaine, presumably, and bupivacaine with adrenaline has got different doses that you're allowed. I presume that it's essentially that levobupivacaine and bupivacaine are treated as the same dose allowance. Is that correct?
Yes, I believe so. I think in the protocol there's a little bit more detail of specifically that, but we can double check that for you. But yes, yeah, we would treat them as effectively the same thing, basically. I'll triple check that and let you know via Chloe. Great, that sounds good from our point of view.
Great, thank you. Any other questions? Nope. If not, that. Oh, was that?
Oh, sorry, just
No,
me.
it's all right.
Just Michelle from Pharmacy. I did send a list of questions to the trial email, so
I think I may have
rather than go.
replied to them this
Oh, have you
morning.
replied?
Did
Oh,
I?
right.
Yeah, sorry.
Okay,
I
that's fine.
think-
I'll have a look.
Yeah.
That's great. Thank you very
Okay.
much. Thanks. I just didn't want to bore everyone with all the research team won't need to know all the things that I've asked. Thank you.
Yeah. Yeah. Have a look. If there's anything, just give me a call or whatever, and we can have a chat about them. But yeah. Great. Yeah. If there's no other questions, we'll sort of write up an SIV report for you with your outstanding actions. Obviously, we'll work through those actions, and once they're all complete and everything's ready to go, you've got your IMP and your pump and everything, then we can look to get you open. So yeah, thank you all for your time today, and we look forward to green lighting you hopefully soon.
Thank you.
Thank
you,
you.
everyone.
Thank
Chloe,
you.
just a few seconds, and I'll just triple check that levobupivacaine thing.
Yeah. Oh, I've got it, I think. Is it this?
Yeah. Yeah, that's it.
Yeah, fine. Yeah.
Yeah.
So. Yeah.
It just, I'm sure I'd seen it before. That's Ben, I think. Well, I'm sure I'd seen it before, but it just on, on the printed out form, one had levo and one didn't.
Right.
So that's fine. I'm sure that was, yeah. Hi, Ben.
Perfect. Well, good to see people are noticing the little details like that. It means you're reading stuff. You're paying attention. Spot the deliberate mistake.
Do you want me to come up? 1 sec. Just give me 1 second. I'll just finish on the call and then I'll come up. Peacock call. Yeah. No. Yeah, that sounds great. Yeah. Cool. See ya. Okay. Bye. Sorry.
Okay. No, fine. We'll let you get away, John. That sounds good. That sounds good.
Great. Thank you very much.
Perfect. Look forward to getting going.
Yeah. Hopefully. Yeah. I'm looking forward to it. Should be good.
Great.
Thank you. Bye.
Cheers, guys. See you soon.
Bye. Bye.